This is a real cancer drug. One mutation makes it useless. Osimertinib treats EGFR lung cancer until the C797S mutation defeats it, and redesigning around that is a multi-year medicinal chemistry campaign. I am Ayush and I built Stanza to run that loop automatically. Hand it a research paper or a UniProt accession and a mutation point, and Claude designs molecules against the resistant target, docks each one into mutant structure, ranks them, and redesigns from the results. And it can trace every number it prints back to where it came from. For this run, I'm handing it a Journal of Medicinal Chemistry paper from December 2025 on beating EGFR drug resistance by targeting a different cysteine, CYS775. Claude reads the whole PDF and you watch it reason in real time. It's not scanning for keywords, it's working out the chemistry, which protein, which mutation, and the hard part, which residue a drug should actually attack. Because here, the residue is not the one that mutated. And there it is. Claude pulled out the target, the mutation, and the residue to attack cysteine 775, not the mutation site. It worked out that C797S destroys the cysteine, the old drug bonded, so the new design has to hit a different one. And every field shows the exact sentence from the paper it came from. Now it builds the structure. Stanza pulls a real experimental structure of EGFR and makes a matched pair, wild type and the mutant, identical except for that one residue. This mutant pocket is what the molecule gets designed against, and cysteine 775 is right there in it, the exact residue the warhead will target. Now the design step. Stanza can seed the loop with proven inhibitors from Campbell, but for the demo I'll only seed it with Claude designed molecules. It proposes 6 molecules and because it knows the target is cysteine 775, everyone carries a covalent warhead, an alkyne, an acrylamide and a cyanoacrylamide. Each is screened by an RD-kit filter and failures are dropped with the reason shown. Now the scoring. Each molecule is docked into both the wild type and the mutant and ranked by composite fitness. Here's the loop. I dock a few, hit generate again and Claude redesigns against those results. One comes back near the top and every score carries an error bar, so a margin smaller than the noise is labeled not resolved and not inflated into a result. For this example there are none. And does the engine actually work? Here's the proof on a completely different target Stanza was never tuned for. BCR-ABL and the T315I mutation that defeats the leukemia drug imatinib. Stanza scores imatinib as defeated and ponatinib, the drug built to survive that mutation, as surviving. With a clean margin, the answer was known in advance and it got it right. So that's Stanza. From a paper to novel covalent molecules, designed, docked, and ranked against a resistance mutation with Claude driving the loop end to end. I am Ayush. Thanks for watching.